All right, so today is going to be a bit of a split lecture. I'd already kind of given you that warning. I'm going to get through, I'd cut out, I don't know if you guys ever look at the slides I post ahead of time, I'm sure some of you do. All that thyroid stuff I'm going to skip. If there's room for it, maybe at the end on Friday I'll chuck it back. I'll go back and visit it, but probably not. We'll see. Today I'm going to talk about the HPA axis, the hypothalamus, the pituitary, and a lot of the downstream targets, and basically the hormone sequence that controls that. And then I will just glancingly talk one slide about the endocrine function of the pancreas, which will set you guys up for your insulin receptor stuff, hopefully. And also after I talk about the GI. And then the second half of the lecture, I'll introduce GI to you guys. So you will get to hear a haiku today. All right. So let's start with the pituitary gland. We will back up the tree to the hypothalamus in a bit, but I'm starting here with the pituitary gland because the pituitary gland is the branch point. It's the branch point in the endocrine system. A lot of things initiate in the hypothalamus, but all of that feeds into the pituitary, and the pituitary is where we get the branching. It is normally when people are being casual in their description of the pituitary, they talk about it having two parts, and I have certainly been casual in my previous mention of the pituitary. I talked about the adenohypophysis. Pardon me as I disrobe in class here on camera. It's hot in here. And then the neurohypophysis. There is a third functional region called the pars intermedia, which I will briefly talk about. It has a single function, which we will talk about just a bit. But histologically, this is what it looks like. And they do have very distinct histological characteristics. So the adenohypophysis receiving hormonal stimulation from the hypothalamus via the hypophyseal portal system, and then the neurohypophysis getting direct neuronal input from the hypothalamus. Okay. Basically, this is an overview here. This neurohypophysis is going to be responsible for the production of antidiuretic hormone. We had talked about that briefly. And also oxytocin. We had mentioned oxytocin, its effect on the uterus and the prostate. And then there's a host of other pituitary hormones that are going to come out of the adenohypophysis. And this pars intermedia is responsible for MSH. And we'll talk about that in a bit. All right. So first, the adenohypophysis and the blood supply that makes it all happen. There are two sort of parallel vascular structures that serve the pituitary. And they are supplied by the superior and then the inferior hypophyseal artery. The superior serving the infundibulum and then the adenohypophysis proper, and then the inferior, just the body of the neurohypophysis. So any of the stimulating hormones, and we'll get to what that even means, the stimulating hormones that are being produced in the hypothalamus are being secreted into this proximal portion of the hepatic portal system here from the superior hypophyseal artery, and then coming down to the adenohypophysis, stimulating that tissue, and then there's the secondary hormone that's going to be released in the adenohypophysis and return to general circulation from that point. All right. So there are essentially three mechanisms that the hypothalamus utilizes to control endocrine function. Two of them I just alluded to, and the third we talked about during the CNS-PNS lectures. Okay, so the first of them is the secretion of various. I'm going to get this out of here. The secretion of various regulatory hormones, and we'll go through those in a little more detail. I'm not going to go in excruciating detail, but a little more detail in a bit. There are various regulatory hormones that are going to be produced by the hypothalamus. They're going to enter that hypophyseal portal system. They're going to enter the blood stream from the superior hypophyseal artery, and then going to come down through the infundibulum to the body of the adenohypophysis, right? It's going to stimulate these the endocrine cells in the adenohypophysis, and then there's going to be some secondary hormonal secretion that will enter the blood supply. So that's the first mode. The second mode of hypothalamic control is going to be the production of ADH and oxytocin. So there's going to be direct neuronal input from the hypothalamus. It's going to come down, stimulate cells in the neurohypophysis, neural because it's being neurologically stimulated, right? And then those cells of the neurohypophysis will produce and release either ADH or oxytocin, depending upon the input that they're getting. Okay? And then the third one is one we had already mentioned, we had already talked about, and this is sympathetic control of the suprarenal medulla. And this is via the sympathetic nerves that are going to descend the spinal cord, emerge along with the splanchnic nerves in the sympathetic, that they're going to pass through the sympathetic chain and emerge as splanchnic nerves, not synapsing in this sympathetic ganglia. And we're going to get these preganglionic fibers that go all the way down to the medulla of the adrenal gland. And this is what's going to stimulate the fight or flight, the production of epinephrine or norepinephrine by the renal medulla. And all of that is getting initiated in the hypothalamus. This diagram is very simplified, simplistic. You know, it's passing on the long course that you can go back and refresh yourself with if you wish. Other questions on that? These are the three general mechanisms. Again, just a broad overview, and we're going to fill in some details here in a moment. All right, so all of these mechanisms are going to have any hormonal pathway that we're going to talk about is going to have some kind of feedback control mechanism. It's going to have some kind of feedback control mechanism. And this is just an outline of what that looks like. So first let's talk about mechanisms in the adenohypophysis, a general pattern that is repeated in many instances depending upon the organ system we're trying to control and regulate. Right? So you're going to have some neurons in the hypothalamus that are stimulated somehow, whether it's by stress or some sort of imbalance, or getting some afferent information from the system, from the sensory neurons or whatever. They're going to be stimulated, and they will release a hormone, some kind of releasing hormone. All right, and sometimes it's called a factor, but usually they're called releasing hormones, releasing factor, whatever. So this releasing hormone is released into what? What is it released into if it's heading towards the adenohypophysis? Into the hypophyseal portal. system, into the blood system, into that portal system. It's a shunt. It's a capillary shunt that we looked at a couple slides ago. This releasing hormone enters the hypophyseal portal system, travels to the adenohypophysis, where there is now going to be the release of some kind of hormone. Hormone number 1, whatever it is. And this hormone number 1 is going to be the hormone that's going to turn on some sort of endocrine organ, downstream endocrine organ. Okay? Because, and this hormone enters the bloodstream, travels systemically, and hits one or more endocrine organs. That endocrine organ is going to produce hormone number 2. It's going to produce hormone number 2. Hormone number 2 is going to then hit a target cell, whatever that downstream effector is, whatever that target is. And also, hormone number 2 is going to have a feedback mechanism. It's going to stop production of hormone number 1 and stop production of releasing hormone. It's going to have an inhibitory effect on the production. So you're going to have some sort of bell-shaped curve, or maybe for you guys it's going this way, a bell-shaped curve in terms of production of all of these 3 hormones. And hormone number 2 is going to be the one that puts the brakes on it. Why do you think it's necessary, looking at the structure that I've given you here, why is it necessary for us to have hormone number 2 and this endocrine hormone? Why can't we have hormone number 1 feeding back on itself? Yeah.
Diffusion? So that it's already near itself, so there could be a little bit and you haven't diffused enough out through the body, but it's still at a high concentration where the feedback receptor is.
Yeah. So number 1, hormone number 1, when it's produced right there, if it's an in situ inhibitor, it's not going to get very far. You see that? Secondly, it doesn't allow hormone number 1 to have multiple target endocrine organs that it can hit. Okay? So that's why we have this multi-stage process where hormone number 2 can then back up and put the brakes on. So what are some examples? We have, here's a list of various releasing hormones that are produced in the hypothalamus. So we have thyrotropin releasing hormone that is going to produce thyroid stimulating hormone that's going to hit the thyroid gland, and then there's going to be T3, T4, the host of various thyroid hormones that are going to get produced, all right, and hit their various target tissues in the body. Some others. Corticotropin releasing hormone is produced in the hypothalamus. It's going to enter the hypophyseal portal system and then affect the cells in the adenohypophysis to release adrenocorticotropin hormone. And that is going to affect the suprarenal cortex. The cortex, not the medulla. The medulla is being directly stimulated by sympathetic nerve fibers from the hypothalamus, as I said in the previous slide. All right, and this is giving us our glucocorticoids, the cortisone derivatives, okay, anything in the cortisone family. Sort of the anti-inflammatory cells of hormones of the body. Then there's the gonadotropin releasing hormone, and this can stimulate the release of follicular stimulating hormone or luteinizing hormone. These have targets in the testes and ovaries and have these downstream release of various androgenic hormones. So that's the general pattern for how feedback control happens in the endocrine system. Oh, the point that I should make here is that all of these hormones here turn off the production of these 2 hormones. Okay, you get that? So everything here that is getting produced because of them is turning off the production of those hormones. You follow me? Everybody? Cool. All right. There are, of course, there are, of course, many exceptions. There are many exceptions. One example of an exception to this is the production of prolactin. So prolactin is the hormone that leads to milk letdown in postpartum mothers and lactating mothers. So we're going to have prolactin releasing hormone, or releasing factor, they call it here, that stimulates the production of prolactin. That prolactin stimulates milk letdown. It also stimulates the production of prolactin inhibitory hormone, and which affects the production of the releasing hormone, not directly itself. So this is not, we don't have that in situ problem that I was talking about with the other system. But it has to go all the way through the systemic circulation to get back up here to both stimulate the production of the inhibitory hormone and inhibit the production of the releasing factor. Okay? You get that? So there's a variation on the pattern. They don't need this intermediary step because there's only one tissue. There's only one target tissue that where prolactin is heading towards. It's not going to have an effect on any other tissue in the body besides its inhibitory effect on its own production in the hypothalamus. And then there's the growth hormone. Yeah?
I guess the way blood flows, is there some prevention of it flowing back through those releasing hormone portal veins?
Yeah, it's not going backwards.
It's not going backwards. It's only going to get back there to block it if it's already diffused through the entire.
Yeah, it's got to go through the whole system. Okay. And you'll notice that it does not have an effect on its own on the cells that are producing prolactin. So prolactin does not affect cells. Yeah, it affects the releasing hormone. All right. So human growth hormone. So there's the human growth hormone, releasing hormone, that is produced in the hypothalamus, which leads to the production of human growth hormone. And human growth hormone has a couple different downstream effects. It goes to the liver. Well, first it goes to all of the cells in your body, the epithelial cells, adipose tissue, liver, whatever. And it's going to affect the proliferation and growth of those tissue types. It's also going to head to the liver and cause in the liver. So this is an effect on hepatocyte proliferation. This is an effect on hepatocyte production of a secondary hormone family of hormones called the somatomedins. And the somatomedins are going to affect all the other cell types that are not encapsulated in this little group over here. So skeletal muscle, cartilage, a vast array of other tissues in the body, right? So in like a growing child. And these somatomedins then do feedback and have an effect only on the releasing hormone, the inhibition of the releasing hormone, and the stimulation of the inhibitory hormone. So there is a sort of balance there between them. Okay? This is a steady state. It's a steady state. Okay. So I think this may be my last. No, I have the one about the pancreas. Yeah. And this would be the slide that I would use to introduce all the downstream targets, and I would then begin to work my way across this and talk about each of these systems, but I'm not going to do that. It's okay, you guys. That's what medical school is for. Anyways, let's start with the suprarenal gland. The suprarenal gland is going to get the direct control. So I said there were 3 methods that the hypothalamus uses to control endocrine function. 1 is via the adenohypophysis, 2 is via the neurohypophysis, and the third one that's very limited but no less important is via the direct sympathetic nerves to the suprarenal gland, right, to the suprarenal medulla. That happens, and we have the fight or flight response from epinephrine and norepinephrine. However, there is adenohypophyseal control of the cortex through the action of adrenocorticotropin hormone. All right, I showed you how it was released in the previous slide, or a couple slides ago, with the releasing hormone. And this ACTH then stimulates the release of the various glucocorticoids. So the corticosterone, cortisol, there's the host of glucocorticoids. And those are going to be released in response to stress. It will have a host of whatever that stress is. Maybe you spend the night camping in the cold, or maybe you are working too hard, worrying unnecessarily about the final in this class, something like that. So you're going to have ACTH stimulation of your cortex. Next is the thyroid stimulating hormone. So all of this is through indirect control via a releasing hormone that is dumped into the hypophyseal portal system that's going to travel and have their effect on whatever hormone that we're talking about here that will be produced by the adenohypophysis. Right? So thyroid stimulating hormone travels to the thyroid, which is right here in your neck, and that's going to lead to the release of T3, T4, the thyroid hormones, which are going to have their downstream effects on the body. Growth hormone travels to the liver, has its own effects on epithelial cells, etc., but it will travel to the liver, and which will then produce the somatomedins, which help you to grow your muscle, bone, etc., cartilage. We talked about prolactin's effect on the mammary glands, follicle-stimulating hormone and luteinizing hormones effects on the testes and ovaries, which have this host of different androgenic hormones that would take a full lecture to even survey. And then here is the pars intermedia. All right, the pars intermedia. Kind of that slide I showed that said there were the 3 methods even sort of overlooks this. Although it kind of gets lumped in, the pars intermedia gets lumped into the same category as the adenohypophysis because it's getting input from the it's getting input via the portal system. It's going to produce melanocyte-stimulating hormone. That is going to lead to the activation of melanocytes, whereby melanocytes, so those are the, you may remember when we talked about epithelium, the skin, the cutaneous membrane, the melanocytes are the cells that are in the basal layer, the papillary layer, or the stratum germinativum of the epidermis. They're going to release little vacuoles of melanin up into, which will merge with the various fibrocytes in the epidermis, and will give your skin protection from the sun, right? So you'll have tanning or whatever happens because of the action of MSH. All right, so this is 1, 2 of the methods of hypothalamic control. The third method is via the neurohypophysis, direct neuronal stimulation of the neurohypophysis by the hypothalamus. Okay, so method 3, and this is it's a method that's limited in scope. So there's either neurons that are going to stimulate the production of antidiuretic hormone. We covered antidiuretic hormone just a little bit in the renal, that one renal lecture that I talked about. And then the production of oxytocin. Oxytocin is effective on the smooth muscle in the ductus deferens and the prostate gland in men during ejaculation. Oxytocin is probably responsible partially for, it is engaged during intercourse for men. And then in women, oxytocin has an effect on the smooth muscle lining of the uterus and the mammary gland. Is released amongst other times during childbirth and helps the woman endure what is often credited as being one of the most painful experiences that humans can endure, which is childbirth. It helps women carry that burden. All right. So there's a flash survey of the downstream targets. Are there questions on this? I'm sure there's probably lots of questions like, what does inhibin do? Why do men and women need it? But we're not going to go there. All right, pancreas. And this is a good one slide to transition to the GI tract. So there are two tissue types in the pancreas. One tissue type is endocrine in nature, and the other is exocrine. The endocrine cells are vastly outnumbered by the exocrine cells. The endocrine cells, which are the ones we're concerned with here, form what are called the islets, the pancreatic islets. You may have heard of the eponym islets of Langerhans. After the histologist who first identified the islets of Langerhans. The islets of Langerhans, and we have one outlined here, is an islet is composed of two cell types. There are alpha cells and beta cells, not very creatively named. The alpha cells are only about 1 in 5, and they produce glucagon. And the beta cells, which are the remainder, 4 out of 5, and they are responsible for the production of insulin. They are ambient glucose reactive, beta cells, production of insulin. So let's, and then these exocrine cells are the pancreatic acini, the acinar cells. And we'll talk about those in the GI chapter just a little bit, okay? And they're responsible for producing pancreatic juice that gets dumped through ducts. You'll remember that's what an exocrine means. It's produced on the surface of the endothelial cell and then goes through a duct and gets dumped into the duodenum, the pancreatic juice, for the digestion of the food. So that's these exocrine cells, the acinar. Anyways, the islets, the endocrine cells, let's look at the homeostasis that happens here. So we have a normal glucose level, and you're anywhere from 70 to 110 milligrams per deciliter. All right. So let's start with the beta cells. You just had breakfast. Blood glucose goes up. This blood glucose goes up, and we get the stimulation of the beta cells, who then produce insulin. What is insulin doing? So there's going to be an increase in the rate of glucose transport into the cells. For those of you who have started on the IR project, you will have come in contact with the GLUT4 translocation to the the surface of the cell, the glucose transporter. That is what this is referring to. It's also going to lead to the increased utilization of glucose and then the production of ATP. This is also something I'm hoping that you will be able to talk about a little bit with the students in your talks. So excess glucogen is then going to be converted into glycogen. It's through a process called glycogenesis. This is going to happen in the liver, skeletal muscles. Those are the two tissues that store glycogen. And insulin basically stimulates anabolic processes. Okay, so the process of building glycogen from glucose that we have, or the process of assimilating protein and not letting it be excreted into the urine, assimilating amino acids and building proteins, and then producing more triglycerides and presumably storing them as adipose tissue. So insulin leads to a whole series of anabolic processes. We see this with carbohydrates, proteins, and fats right here. All right. So that's this slide could be a nice overview kind of thing for those students, and then you would talk about each of these in a little bit more detail with them. And then that's going to ultimately lead to a reduction in blood glucose level, and we'll come back to homeostasis. All right, so you had your breakfast, we released some insulin, we utilized the glucose, stabilized the blood sugar, and now it's wearing on. Time is wearing on, and the blood glucose begins to dip. The blood glucose begins to dip. So the alpha cells are going to produce glucagon. So glucagon in general stimulates catabolic processes, catabolism, breakdown processes. So we're going to have glycogenolysis, the breaking down of glycogen. And that's going to happen in the liver and the skeletal muscles, because that's the only place that we're storing glycogen, liver and skeletal muscles. And then again, the breakdown of fatty acids, and then the synthesis and release of glucose that will happen in the liver. All right, and we call that gluconeogenesis. Gluconeogenesis, the production of new glucose. Production, genesis, new glucose. Gluconeogenesis. That then will raise the blood sugar level to keep your blood sugar nice and steady. Okay, so this could happen, for example, when you're jogging. You're jogging. You need some. You've got your glycogen has been stored in the liver and the muscles. You're burning glycogen, but you're taking a nice long jog, and you've gone 45 minutes, and you need to keep that blood sugar up and stable. Glucagon is doing that. Okay, so that's all of that material on the endocrine that I'm going to give you guys. I just did the pancreatic part because seems like that is going to be some useful stuff for you guys. Are there questions on that before I begin this? No? Okay. The last haiku of the semester. Here we are. We are one long tube, parsing nutrients and filth from mouth to anus. How charming is poetry. All right. So the GI tract, people, not to get too corny with the pun, tend to look down on the GI tract. Here is a statement by this guy, Alessandro Benedetti, in 1497, 5 years after Columbus sailed the ocean blue. His ruminations on the GI tract, quote, "The stomach is the lowest and has a hidden place in the body because of its uncleanness, as though nature had spared the principal members and had relegated the stomach or bowels farther away from the sight of reason and of the mind, and fenced it off with the diaphragm in order not to disturb the rational part of the mind with its importunity. These members serve the higher ones. Some of them concoct the food into juice, others digest it into various humors, others expel the superfluity." I clearly never changed a diaper. So, despite this gentleman's clear aversion to the alimentary, the less savory aspects of the alimentary canal, he did allude to some of the interesting elements of it, so that there are organs that concoct the food into juice. This is alluding to the fact that we are going to liquefy, emulsify our food. We're going to masticate it, grind it up, and then suspend it into solution. Others digest it into various humors. I'm assuming that he's talking about farting. And then expel the superfluity. So when he says superfluity, this hints at the fact that there are aspects of food that are required, and then there are, of course, the superfluid elements which the body excretes. But there are things in food that are necessary for life. Da Vinci had this to say. He believed that it helped the respiratory system in its function. The compressed intestines, oh, this is funny, yeah. The compressed intestines with the condensed air which is generated in them thrust the diaphragm upward. The diaphragm compresses the lungs and expresses the air. I'm not sure whether he's talking about belching or farting here, but he clearly had some kind of gas. Anyways, Benedetti, he was at Padua, the University of Padua, and here's Da Vinci's depictions of the GI tract. Interestingly, he has some pretty nice descriptions of the abdominal vasculature. All right. Oh, did I mean to show this slide? I feel like I was going to... Okay. This is the wrong PowerPoint. I had meant to cut a lot of this out, I think. Well, we'll just run with it. So, peristalsis is the process by which food is moved through the GI tract. Our GI tract is essentially a long muscular tube. It's a long muscular tube. And we chew food up, it becomes a bolus, and we're going to amend that bolus at different points. We move that bolus by a rippling wave of contraction that happens upstream of the food and pushes it downstream. So this wave of musculature behind whatever bolus, moving it along the GI tract. And here we see how this works when we swallow. There's the buccal phase, or chewing things. Then you begin the pharyngeal phase, where swallowing it clamps down on the epiglottis, and this peristalsis happens that drops the food down into the stomach. So one thing I will point out here in the mouth is we have these salivary glands, and here's a cross-section of what looks like the submandibular gland. And these salivary glands are for the production of amylase. Amylase is an enzyme whose job is to clip glucose molecules off of a starch. All right? So it's going to take a starch, which is basically the plant version of glycogen. Amylose is the plant version of glycogen. It's starch. And it's amylase is going to clip sugars off the end of the branch points in amylose. All right, and this starts in the mouth. This starts in the mouth. There's various layers of the digestive tract. There's a longitudinal, a circumferential, and sometimes an oblique layer of muscle. Between these various layers of muscle, this is the muscularis mucosa here, this muscle layer. There's these various layers of smooth muscle. In between them are these vast networks that we call plexuses. You've heard that term. And specifically here, the enteric plexus is the neural web that controls the GI tract. Okay? It has some connection with the central nervous system via vagal stimulation, but in general, it's pretty autonomous. It's pretty autonomous. There are these enteric reflexes that don't head towards any central location. Right? You don't think about moving a bolus of partially digested food through your intestines. It's just sitting at one point in the intestines. The stretch receptors in the intestines sense that and then will initiate peristalsis via this myenteric plexus. So the submucosal plexus talks to the myenteric plexus, and there's this small feedback loop that leads to peristalsis and the propulsion of food through the GI tract. Does that make sense, everybody? Sort of? No? It's pretty simple. Oh, here's the picture. Anyways. So there's three general ways that this is regulated. The first is a neural mechanism that does connect with the central nervous system. They call this the long reflex. All right. So there's going to be various stretch receptors, chemoreceptors that are somewhere in wherever we're talking about in the digestive tract. There is going to be some sort of afferent signal sent to the central nervous system, and then that's going to send some efferent output along the vagus that will help stimulate the myenteric plexus. That's going to this myenteric plexus will trigger peristalsis, segmentation movements. So you have this big dinner, and you're going to like break it up into manageable bits as it's moving through the GI tract, and then it's going to feed into these other mechanisms. So the second mechanism is endocrine in nature. There's these various hormonal mechanisms that are going to help regulate digestion. There's going to be some kind of hormone that is, well, hormones are going to be released that via circulation are going to lead to secretory cells releasing some kind of factor. And in the stomach, for example, that would be like gastrin or by the G cells or whatever, the parietal cells and HCl, whatever. Any of these different secretory cells are going to make buffers, acids, whatever kind of enzyme is necessary for digestion. And then third are these local factors that are, there's these various local factors, the pH, physical stimulation of having food in the area, chemical stimulation. These local factors are also going to drive secretory cells and the enteroendocrine cells, which are producing these hormones that are feeding back and affecting the secretory cells. So those are the three overarching mechanisms. Why don't we? Well, we got a couple minutes. Yeah, I can start this. Here's the stomach, and let's just go over a little bit of the anatomy so we can pick up next time and talk about the physiology. The lining of the stomach has what are called rugae, these rugae. And if you were to turn a stomach inside out, sometimes I have a picture of one. There are these ridges. It looks a little bit like the surface of the brain, actually, like the gyri and sulci of the brain. So in the stomach, they're called rugae. And down between each of these rugae, we have what we call a gastric pit. A gastric pit. No, that is not a greasy spoon. It is an anatomical feature of your stomach. These gastric pits are lined with mucus cells. The job of the mucus cell is to produce mucus to help liquidify the food that is in there, to help make it nice and slimy so it'll pass through the GI tract unimpeded. And then descending from these gastric pits are gastric glands, and we see them here. And in the gastric gland, there are a host of various cells. We're going to have parietal cells, G cells, and chief cells. We'll talk about what they all do next time. Yeah, let's leave it at that. Are there any questions for what I talked about today? So, what did I talk about today? I started in this whirlwind lecture back in the endocrine system, talking about the action of the hypothalamus and the hypophysis or pituitary gland, the feedback control mechanisms that we use with the releasing hormones that then cause the release of primary hormones, which hit a target organ, to the secondary hormones, and then the feedback inhibition that secondary hormone has. We talked about some variations on that theme with prolactin and growth hormone. Then I talked about a little bit about, I just introduced the different downstream target organs and some of the associated hormones, and talked about the endocrine function of insulin and glucagon, the homeostasis of blood sugar. And then I talked about peristalsis here a little bit and introduced some of the anatomy of the GI tract. Yeah, Becky?
I thought that next week we were going to do the A groups.
Definitely, absolutely. The last lecture in this class is Friday. Yeah, what's confusing?
We don't have class on Friday.
Oh, there's no class this Friday?
Because of the COVID.
Yeah, sorry about that. Okay. Well, there's the GI tract for you, folks. What a great semester. Yeah, pardon me for not being fully integrated into the Colby schedule. They don't include me in a lot of those emails. I guess I'm not. Right, Colby Liberal Arts Symposium Friday. Enjoy that. I've enjoyed lecturing to you guys. Thank you. So the GI tract, you're not going to be responsible for a whole lot here. There you go. Are there any questions? Thank you for telling me that.